Protein sequencing is a practical process of determining amino acid sequence to identify protein or peptide or to characterize its post-translational modification. So there are several steps. First step including breaking of disulfide bond. Then there is cleavage of polypeptide chain. And then third step is sequencing. There is sequencing of our peptide that we want to know the amino acid sequence, and then ordering of that fragment of the peptide which we have cleaved. Then fifth step is locating the disulfide bonds in the peptide. Then there is two steps in breaking of disulfide bond, that is first is oxidation by formic acid, and then there is reduction step with the help of DTT or beta mercaptoethanol, and cleavage is done by proteases which cleaves the protein and peptide bonds. And then sequencing will include three steps again. First can be to identify the N-terminal amino acid, and then second is the most important one, that is the Edmonds degradation, and then third is to identify C-terminal amino acid. Then we have cleaved amino acid which can be ordered, or the fragment of amino acid which can be ordered by overlapping the fragment, because different proteases have cut it at different sites. So overlapping can be used to deduce that sequence, and then electrophoresis can be done to locate the disulfide bonds. So we have studied 5 basic steps of the protein sequencing. Now we will see one by one, starting from the stage 1 or the step 1. So first is breaking of disulfide bond. Now why does disulfide bond is need to be broken before the cleavage of polypeptide? Why it is not done directly? We are not cleaving directly the peptide bond, because there is a reason that disulfide bond interfere with the sequencing, and to avoid this problem, this disulfide bond should be cleaved out, because it do not allow the release of amino acid from the peptide, so it must be cleaved. So for this we have already discussed the 2 steps, that is we have 2 approaches to do or to cleave the disulfide bond. That is first is oxidation step and the second is reduction step. So oxidation will include or done by formic acid, and which is a product of formic acid and H2O2, the reaction of formic acid and H2O2. So it will cleave the disulfide bond of polypeptide and give 2 cysteine residues. In the same way, reduction is done by reducing agents like DDT, dithioethol, or beta mercaptoethanol can also be done by beta mercaptoethanol, which gives 2 cysteine residues. But in presence of iodoacetate, it also prevents the formation of disulfide bond. So here is the reaction in which this 2 cysteine residues, we will get 2 cysteine residues by reduction of peptide with the help of beta mercaptoethanol or by DTT, and this is the cysteine residue will be converted into carboxy methylcysteine by treating it with iodoacetate. So this carboxy methylcysteine has a special function which can stop the disulfide bond formation. So we have so there is a breaking of a disulfide bond. Moving on to the next stage, there is, after breaking disulfide bond, there is a cleavage of polypeptide chain, and we know that this is done by proteases, and we can also use some chemical agents. And these proteases or chemical agents have different targets which we are going to study. Like first can be, it can cleave carboxylic acid or carboxylic side. It can cleave amino side of the peptide or amino acid, the amino side of the peptide, and carboxy peptidase are there, and then there is a hydroxylamine which can be used to cut the peptide or the protein. So let's start with the cleavage of carboxyl group. So these proteases cleave after the various amino acids, like there is Kr, which is lysine. K stands for lysine, and R arginine, which is by trypsin. Cleavage is done by trypsin after these amino acids. And then there is phenylalanine, tyrosine, tryptophan, FYW by chymotrypsin. Then GAS is glycine, alanine, serine by elastase. And then methionine by cyanogen bromide. Then there is arginine alone by thrombin. This is thrombin. Thermolysin will cleave the NH2 bond. Sorry. And then there is tryptophan, which get cleaved by orthoiodosobenzoate. And the trypsin, chymotrypsin, and elastase, there is one condition that there should not be a presence of proline after these amino acids, which they specifically cut. So moving on to our cleavage of amino group, which is done on valine, leucine, isoleucine, and phenylalanine by thermolysin, and then leucine, phenylalanine, tyrosine, tryptophan by pepsin. And there is also condition for the pepsin that proline should not be present before these amino acid that pepsin specifically cleave. And then carboxy peptidase, we know that it is of two types, that is carboxy peptidase A and carboxy peptidase B. So A will cut the C terminal of the aliphatic or aromatic amino acids, except arginine and lysine. K is lysine and R for arginine, and also it do not cut proline as well. And carboxy peptidase B cuts at the C terminal of basic amino acids, that is lysine and arginine, opposite of that of A. And hydroxylamine has a different kind of cut, like it cleaves the asparagine and glycine bonds. So we have studied the cleavage of polypeptide chain. This was the cleavage of polypeptide chain. So till now we have studied two stages or two steps by which protein sequencing will be further done, and next step will be discussed in next videos. Thank you.